Hey everyone, and welcome to our video on investigating tropisms. This is part of our Module 5 Part 5 series looking at those plant and animal responses, and what we're going to be doing in this video is having a look at specification reference 5.1.5a Part 2, Practical Investigations into Phototropism and Geotropism, and 5.1.5e, Practical Investigations into the Effect of Plant Hormones on Growth. So let's start off with phototropism first of all then. Now, phototropism is something that scientists have been investigating in plants for quite some time. As we can see, here's an experiment back in 1880. And while we don't have to recall exact details of these experiments themselves, you could get questions on the exam paper related to these sort of experiments and asking you to interpret or analyse results from it, etc. So what we're going to do is go through the variety of experiments that have been done on phototropism, and then we will just talk about some of those practical considerations that you should have as far as experiments you could have done in class and therefore could be asked about on your exam to do with phototropism as well. So in 1880 we had Darwin's experiment, and quite simply what this experiment consisted of was taking our little coleoptile and then removing the tip of it. So what we can see at the bottom is our little summary of what happened. So if we had the full coleoptile and we shone light from one side, so unidirectional light, uni obviously meaning one, directional direction, then what we found is that the full coleoptile would then bend towards the light. In the instance where we'd removed the little tip of the plant, then it didn't bend, it just grew vertically. So obviously we could have that suggestion that this might be down to the wounding of the plant when we're cutting that tip off. Therefore, rather than cutting the tip, we just covered it with an opaque cover, so no light was able to reach that tip. And exactly the same thing happened there. It just grew vertically, regardless of that unidirectional light. So from this, Darwin concluded that the tip was responsible for detecting the light that then led to this bending of the actual shoots. What we now know is it is good old auxins that are responsible for this phototropism. And what we find is that we've had a whole range of experiments carried out on plants using auxins to dig deeper and understand exactly how they have their effect. So the next one, 1913, Boyce and Jensen's experiment. We're back to our little coleoptiles again. And we've cut the tip off once more because that seems to be the starting point for a lot of these experiments. But this time, what Boyce and Jensen did was they actually put this little barrier in there of gelatine. Now, what they then did was place the little tip on top of that gelatine barrier, and what we then saw, it bent towards the light again. So from this, they came up with the conclusion that there was some kind of a chemical being produced in the tip that caused this response, because that chemical was able to diffuse through the gelatine and reach the rest of the stem. So they followed it up with a second experiment, and what we then have is this little insertion of something called mica. Now mica is impermeable to chemicals, so this time what he did was he placed the mica barrier on one side of it, the shady side, but only halfway through that coleoptile. And then he did the same thing with the barrier on the lit side. So what we could then observe by having that unidirectional light is that when the mica barrier was placed on the lit side, then the plant still bent. When it was placed on the shaded side, it didn't, it grew vertically. So from this, we can actually conclude that the chemical that is being produced in the tip is diffusing down the shaded side of the plant, because if mica is a barrier to chemicals and on the shaded side it doesn't bend, clearly it's stopping a chemical moving down that side of the plant. So what we do know is that we have got something that is then diffusing down this plant and it's stimulating growth on the shaded side. It's not something being produced that then inhibits growth on the lit side. And that mica barrier experiment just proved that to us. So at the end of Boyce and Jensen's experiment, we knew that there was a chemical that travelled down the shaded side, stimulating the growth of the cells on the shaded side of the plant. 1919, we come to Powell's experiment, and Powell cut the tips off the coleoptiles, but then replaced them off-centre. Now, whenever the tip was replaced off-centre, whichever side it was placed on, that one grew more than the side it wasn't on. So this again backs up the idea that there's something moving through that plant tissue that stimulates the growth. Went is the final one that we're going to look at in 1926, and again we're cutting the tip off the coleoptile, but this time placing on little gelatine blocks which absorb that chemical, the hormone. And when he then placed the gelatine block back onto the coleoptile, it caused that curvature. And he then followed up by adding a few blocks, and therefore he concluded that the degree of curvature shows correlation to the number of those tips used. So the more of those blocks we put on, then the greater the degree of curvature. So we can then conclude that the greater the quantity of this particular chemical, then the greater the degree of curvature will be in our plant. So one of the things that we need to understand then are the practicals that you could have been doing in your lab setting in terms of phototropism. And one of the questions that does come up quite frequently in regards to those practical skills is a control. So if you're doing a phototropism experiment, you would need your control alongside your test subjects. And the control for phototropism, quite simply, is the plant that's going to be illuminated from all directions. Because phototropism is all to do with obviously that unidirectional light and causing it to bend, what we see for our control is a sample that must have the light from all directions. So just remember, your control will be illuminated from all directions, your test plant illuminated from one side only. Now, what we may well find here in terms of the experiments they could talk to us about in terms of the procedure, then a serial dilution might come in here. And this question does come up from time to time, and there's a couple of points that you should be mindful of. First of all, when you are talking about a serial dilution, then we need to make sure that we use the phrase distilled water. So you are going to take a measure, and obviously depending on how much you're diluting it, that will determine the actual volume. But we are going to take a volume of our stock solution, and then we are going to add that to another volume of distilled water. So please get in the habit of writing that we are adding distilled water, not just water. So that's our first point, is that we are going to be using distilled water, not just water. Second thing is to make sure that you talk about mixing before you take the next sample, because remember, in a serial dilution what we're going to have is our first tube, which might be our 100 parts per million, then we're going to be diluting down to 10 and to 1, etc. And what we're going to be doing is taking from the previous tube in each case. So in this example, what we'd be doing is taking 1 centimetre cubed of our previous solution and adding it to 9 centimetres cubed of distilled water in each case. So we need to mix that tube after we've added, before we then take the next sample out. So make sure you talk about mixing. And of course, we need to then make sure that we are working in that logical sequence, and we refer to taking the sample from the previous dilution. If you do that, you should be able to get maximum marks when describing how to do your serial dilution. The only bit that you might have to do is obviously work out the volumes of the distilled water and your stock solution that you'd be using, but we look at that in another video that focuses more on the actual practical skills. So we have said that this auxin causes the elongation of cells. What we just need to have a little look at is how it does this. So auxin is obviously a hormone produced in the tip of our actual shoots, and it diffuses down that shaded side. Now, when it comes into contact with those cells on the shaded side, what it's going to do is actually promote the transport of hydrogen ions by ATPase. What that's going to lead to is a low pH, and that is the optimum pH for these things called expansins. Now, expansins are, as the name suggests, something that can expand, And therefore, what's going to happen is those expansins break the bonds within the cellulose, which is obviously within the cell wall of our plant cells. And because we've broken the bonds within the cellulose, the cell walls are now a little bit more flexible. They're less rigid than they were. As a result of that, water can enter the cell more readily, and so it expands. The other type of tropism that you could be asked how you would investigate it in a lab setting is geotropism. Now, geotropism is all to do with the plant's response to gravity, and in order to investigate this, we use a bit of equipment called a clinostat, which we can see at the bottom here. Now, clinostats are probably very high-tech in your school labs, if they're anything like mine, where you may well just have a plastic beaker stuck here onto the arm of a device connected onto a little motor that spins. Now, the whole idea is that we're going to place our plant within the plastic beaker, and it's going to be horizontal. And if we're talking about the control, then we turn the clinostat on. So basically our clinostat is then going to be rotating very slowly. We're not talking about some kind of fairground ride where it's flinging plants across the room with G-forces. Instead, it's this gentle rotation, but what that does is it actually applies the gravity equally to all sides because the plant is just rotating continuously. As a result of that, that plant is just going to grow horizontally. The test plant has to have the same setup, so we still have to put it in the clinostat, but it's switched off, so it's not rotating in that case. And in those instances, we will see the bending of our plant in response to gravity. So just remember, when it comes to geotropism, the clinostat is switched on as a control and switched off for the test plant. Go careful not to mix those two up. So the last thing to bear in mind here is that when we are talking about auxin, it has different effects in the shoots and the roots. So if we have auxin in cells of our shoot, that is going to stimulate cell elongation. If we have auxin in the cells of our roots, it inhibits cell elongation. And the reason that's important is when we're considering our geotropism, when we've got our little root here, then the auxin is going to be accumulating on that lower side, the side closer to the ground, because of gravity. And because it's going to be inhibiting cell elongation in that lower side of the actual root, the side closest to the ground, it causes the whole root to bend downwards, i.e. it makes it bend towards the ground so that hopefully that seed will then have its roots in the soil. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when the next video is uploaded, and of course head on over to the website where you can find a range of other resources to help you in your A-level biology studies.